bodies contributed to the body's defenses. In today's lecture, I will talk about the actual cells and of the innate immune system, and also their functions and how they behave or how they recognize that targets like bacteria, viruses don't belong to the body, what we call non-cell. So as we covered yesterday, the term innate refers to inborn. So as soon as we come into this world, this is what our DNA codes. So it's invariant. It's not able to adapt or change. It's the first line of defense, but it's quite limited as opposed to the potency of antibodies or T lymphocytes. But nevertheless, even though we make this delineation of first line, unchanging, innate immunity, always bear in mind that the two arms, the innate and the adaptive, are in very close communication with each other. The innate, because it is the first line of defense, serves like a security guard to detect any changes or any threats in the body, to start off this inflammatory process, but then provide signals to draw in the cells of the adaptive immune response, like B lymphocytes and T lymphocytes. And an important point to note also is that these innate immune cells play normal homeostatic functions to maintain a healthy body. They remove dead or dying cells, they remove any debris, because all the proteins, all the components in our body have a certain use by date, and they degrade over time, so they have to be removed. So if we classify all the different white blood cells or leukocytes in the blood, we can group them based on their size and their shape. So the largest cells and the heaviest cells in the blood, we call these granulocytes because they contain dense granules if you look at them under the microscope. These granulocytes are composed of neutrophils, eosinophils, and basophils. Then the next big important set of cells are monocyte macrophages. These two terms refer to the same thing, except that monocytes are more immature macrophages, and so you find them circulating in the blood. If you take a blood smear, you will identify monocytes. When they get activated, we call these macrophages. And the thing about the monocyte macrophages is that they are a wide continuum of cells, all at different stages, all its different functions. So we also have tissue-fixed macrophages. So these are specialized macrophages which don't migrate in the blood. And so we have macrophages which are in the liver, which we call Kupffer cells. In the brain, we call these microglial cells. In the lung, we have alveolar macrophages, and so on. The next group of cells are probably the most important for communicating with the adaptive immune system. And these are dendritic cells. Their function is very similar to macrophages, but their specialized function is to present antigen to T cells, which you'll learn about in a later lecture. And then finally, we have the natural killer cells, which are specialized in recognizing targets like virus-infected cells, like tumor cells, and then sticking to them and directly killing them to prevent more virus infection or more progression of the tumor. This is the distribution of these different cells if you looked in a normal blood sphere. And you can see that by far the greatest number of cells you find in the blood are neutrophils. And this reflects their importance as a first line of defense as soon as you're infected with bacteria, to come capture all of that bacteria and then prevent it from spreading further in the body, and then also drawing in the other cells. Then we all have eosinophils, which I won't talk about until later on in this course, because eosinophils are more specialized with reference to allergic reaction. Basophils are important in terms of the inflammatory response, but for 60% of this course, the important cells that you learn about are monocytes and dendritic cells. If we group these innate cells based on function, we can group the functions as phagocytosis, cytotoxicity, activation of adaptive immunity, and hypersensitive reactions, or generation of an inflammatory response. So phagocytosis, you all understand what the term phagocytosis means? Give me a definition. Engulfment, eating, capture, right? Cytotoxicity is obvious. Activation of adaptive immunity, this is brought about through the production of cytokines, these protein messengers, which you'll learn about in another lecture, but also in the expression of particular receptors, which are then taken up by lymphocytes and other cells. Hypersensitivity, as I said, we'll cover in one of the later lectures. This is a video, a time-lapse video of a neutrophil chasing these bacteria. And then engulfing it. This is the process of phagocytosis. Now, if we break down the sequence of events that you see there, we can see there are different steps. In the first, the macrophage or the neutrophil has to identify that bacterial cell as being a target to phagocytose. That is dependent on receptor ligand interactions. The ligand being the so-called antigen on the bacterial cell, the receptor on the phagocyte recognizing that ligand as being foreign or non-self. So you need this receptor ligand interaction initially. Then once that binding happens between the phagocyte and the bacterial cell, you get changes within the cytoplasm of the phagocyte to change its actin myosin filaments so that the cytoplasm can swamp or cover over the bacterial cell to draw it inside. That bacterial cell then gets drawn into a vacuole within the cytoplasm, and this is called a phagosome. Meanwhile, within the cytoplasm of these cells, particularly in neutrophils, you have these dense granules, and these granules are packed with proteolytic enzymes, defensins, antibacterial chemicals, which, when you get fusion between these lysosomes and the phagosome to form what's called a phagolysosome, these packets of enzymes are discharged into the phagosome, which can then act on this ingested material and degrade the bacterial membrane to break it down into various components. And then in this diagram, these broken down fragments of this ingested bacterial cell are then excreted. But there's another important component of that, is that this phagosome keeps these degraded fragments and presents them to T cells, which we'll learn later in another lecture. So, how did that neutrophil, that phagocyte, recognize the bacteria as being foreign? What tags or what markers are displayed on that bacterial cell which we don't display in our own bodies? These are called pattern-associated molecular patterns. These are the antigens which are recognized by cells of the innate immune system. They're called patterns because these particular molecules, which are recognized by these receptors, consist of repeat units of complex lipids and sugars. Where would you find complex lipids and sugars in a bacterial cell? DNA is one of them. On the cell membrane, cell wall, bacterial cell membranes are made up of repeat units of complex sugars like lipopolysaccharides, teichoic acid, and so on. So unlike the target antigen that antibodies recognize, which are proteins, which are highly variable because it's dependent on the amino acid sequence of the protein, this system recognizes repeat long chains of sugars and lipids. And so this is why we call them patterns. So this group of antigens, which you call PAMPs, we can divide them into antigens or PAMPs which are associated with microbes, whether they're bacteria, viruses, or fungi. So during an infection. And so we call these microbial associated molecular patterns, or MAPs. And these, because they're lipids and sugars, you can find them on bacterial cell walls, fungal cell walls, but also importantly, nucleic acids, because cells don't use double-stranded RNA. But a virus like COVID-19 goes through a double-stranded RNA intermediate. And so this is the next point that you need to understand, is that cells themselves have an internal immune system. The inside of a cell can detect what is foreign and belongs to the cell and what isn't and should be dealt with. And this is usually at the level of the nucleic acid. Bacterial cells have a particular nucleotide motif called CpG elements. Normal eukaryotic cells don't have that. So as soon as these elements appear, the cell gets activated to eat itself, to eat its inside. The other thing to note is, in keeping with this theme that I'm telling you, that the immune system is also there as a homeostatic mechanism. The other type of antigens recognized by these innate receptors are these so-called damage-associated molecular patterns. And so this is similar to, or this is pertinent in the description I gave you yesterday when you get sunburn. When you get UV rays necrosing, killing cells from too much UV exposure, the release of those intracellular contents is detected by these receptors from surrounding healthy cells. They pick up these intracellular contents like cellular DNA, mitochondria, heat shock proteins, and whatever, and get activated as a result of picking up these components to generate an inflammatory response. This is why you get heat, redness, swelling, pain during sun. And you also get the same sort of reaction if you cut your finger or stand on a nail or whatever. The receptors that recognize these pumps are called pattern recognition receptors. And there's three main classes, which are called toll-like receptors, nod-like receptors, and rig-like receptors. The toll-like receptors were one of the first to be discovered, and they were discovered by three independent groups. One group that was looking at the immune system in mice, another group that was working on plants, and another group that was working on worms. So this system, we can find the DNA coding for these genes in things as diverse as plants to lower organisms like sponges, worms. And this shows how this system has evolved with life in terms of detecting what belongs to the body or self versus what is in the extracellular environment or non-self. And these receptors can either be on the surface of our cells or inside the cell, within the cytoplasm, or within vacuoles. Or they can be secreted and float around like specific ligands to capture any of these PAMPs, these floating sugars or whatever. So the biggest group of toll-like receptors, biggest group of PRRs are the toll-like receptors. And there's something like 14 different species of TLRs. Some of them are present on the surface, some of them are present within the membranes of intracellular vacuoles. One common one is TLR4, which you'll learn about in microbiology when you understand that TLR4 recognizes lipopolysaccharide of gram-negative bacteria, particularly E. coli. I don't know how much microbiology you know at the moment, but have any of you heard of endotoxic shock? Okay, so endotoxic shock, an endotoxin is, you know what a toxin is. So an endotoxin is a component of gram-negative bacteria, particularly LPS, this lipopolysaccharide. When you have a severe infection with gram-negative bacteria, you get massive amounts of this LPS being released. This LPS gets recognized by this toll-like receptor 4 on macrophages. And what it does is it stimulates a huge inflammatory response by macrophages so that these inflammatory cytokines go systemic. They pass through the body in the blood. And there's such a huge generation of inflammation that it causes blood to clot, blood vessels to get damaged, and basically your organs don't get enough blood, they don't get enough get enough oxygen, and you can die from it. So the consequences of these pattern recognition receptors recognizing the target, these pumps, what that results in is basic biochemistry, is that once the receptor gets engaged, you get a what we call signal transduction, or a transfer of chemical energy involving phosphorylation, dephosphorylation, going from that receptor into the cell to the nucleus. Once the signal gets relayed into the nucleus, that will activate basic transcription factors, which will unwind your chromosomes and expose particular genes. These genes will then get transcribed into protein, and these proteins can be involved in cytokine production. They can be involved in instructing the cells to divide, to be more phagocytic, and so on. This is basically how this whole immune system works, through this engagement of receptors and then passing of the chemical signal into the nucleus to cause changes in the transcriptional program. Another effect, or the result of PRR engagement, concerns this particular group of receptors, the nod-like receptors. Now, there are certain enzymes in the body which are synthesized but remain in the cells as latent forms. They're composed of a fragment of the protein which, think of it like bubble wrap, keeps the enzyme non-functional. Once that inactive component is cleaved off, it releases an active enzyme. When nod-like receptors detect viral double-stranded RNA, they aggregate into this multi-enzyme complex called an inflammasome. The target of this inflammasome are two of these inactive cytokines. One is called IL-1, or interleukin-1, which you'll learn about later. Another one which is called interleukin-18. These are potent inflammatory cytokines. So when these nod-like receptors are engaged as a result of the presence of a viral infection, these inflammasomes then cleave or activate the pre-existing IL-1, IL-18 in these macrophages, which can then be released and then activate neutrophils to come into the inflammatory site, activate T cells, B cells, and so on. So, as I said, the neutrophils are the most important in respect to this frontline defense because they're so abundant. And in fact, we're seeing that a lot of these COVID fatalities are due to people who are not able to mount a good neutrophil in the initial infection stage, which shows how important they are. The thing about neutrophils, as you can see, they contain all of these granules that I mentioned. That when this neutrophil has phagocytosed a target and the granules are released, these granules themselves not only kill the ingested target in the phagolysosome, but also kills the neutrophil itself. So the neutrophil then bursts and releases its DNA, its intracellular contents like mitochondria, heat shock proteins, and so on. One of the functions of this is that DNA is quite a viscous substance, like glue. So when these neutrophils burst and release their contents, they provide a sticky matrix to trap any bacteria that are there to prevent them from spreading further in the body. But the other function too is that these intracellular contents are then picked up by the PRRs of surrounding healthy cells to recognize that there's cellular damage there, and so causes them to create inflammasomes, generate inflammatory cytokines, and so on. So macrophages are like generals of the immune system because they have different functions. Not only can they phagocytose and trap foreign particles, but they're also an important source of these cytokines. So once their PRRs are engaged and they become activated, they will become more phagocytic and take up more bacterial cells or viruses or whatever. But also they will be activated in expressing surface molecules to activate nearby lymphocytes, as well as producing large amounts of cytokines to draw in or amplify an immune response. But then, as we'll learn later, we have two classes of macrophages. The macrophage that's involved in an inflammatory response, we call these M1 macrophages. The macrophages which are involved in normal homeostasis, like vacuum cleaners, just to suck up dead or dying cells, but importantly, not to generate inflammatory response. These we call M2 macrophages. And these M2 macrophages, because their role is just to remove cells and not to generate an inflammatory response, they produce cytokines which inhibit or prevent these active cell-damaging immune cells to come into action. So these M2 macrophages play a role in fetal development because when after a few days of gestation, the fetus undergoes various morphological changes. A lot of the cells between the fingers go into apoptosis. All of these are removed by these so-called M2 macrophages. So these macrophages have a role in an inflammatory response, but also in non-inflammatory responses too. And then these specialized cells, dendritic cells, are similar to macrophages in that their main function is to phagocytose antigens. They're called dendritic cells because they have these long processes, like dendrites, that you'll find in the brain, in neural cells. These long processes are actually increase the surface area of the cell, right? But if you had to look with a powerful electron microscope, you'd see that all along the surface, the more surface you have, the more receptor expression you have. And these receptors are providing antigens to be recognized by T lymphocytes and B lymphocytes. So this is the role of dendritic cells, to take up foreign non-self antigens and then process them into small fragments of a few amino acids to be presented or shown to T lymphocytes through the receptors. So this is why they are called professional antigen presenting cells. We'll talk about that in another lecture. But all of these cells, whether their function is to phagocytose, whether it's to be cytotoxic, their other role is to make these important proteins called cytokines, which act in communicating the whole immune system, to bring in the adaptive, to bring in the more neutrophils to the side to generate inflammation or whatever. So finally, we'll talk about these professional killers. Stop music. So I'll talk about natural killer cells later, but I just want to mention a particular theme in immunology, that natural killer cells, again, they don't recognize PAMPs like macrophages and neutrophils. What they recognize is a dual recognition system. There is all the cells in our body express a particular surface protein which we call HLA or MHC. The expression of this HLA or MHC determines whether we can accept or reject a transplanted kidney or a liver from other donors. The closer your HLA match is, the more chance you have of accepting that foreign tissue in your body. Now, natural killer cells, every day, they are scanning all the cells in our body. And where they see a healthy cell expressing this healthy HLA molecule, it instructs the natural killer cell to leave that cell alone because it's part of the cell. When you get a tumor cell developing, when you get a virus-infected cell, that HLA gets changed, gets modified. And so the natural killer cell will see that and identify that as a target to be killed. So basically what the natural killer cell is getting is a mixture of kill me, don't kill me signal. Because if you imagine a cell that's just been newly infected with a virus, is going to start expressing some of this virus antigen on the HLA, but there'll also be normal HLA as well. So the natural killer cells get a combination of danger, normal signals. Whichever is greater will determine whether that natural killer cell then kills that cell or leaves it alone. And this theme will also, you'll also find out when we talk about T cell activation that these immune cells work or are controlled by a combination of positive and negative signals. And whichever is greater will determine whether the immune system becomes an inflammatory response or a non-inflammatory response, and so on. Okay, that's it. Any questions?